Hello everyone, I'm Mr. Furlong, and today we're going to talk about cell transport part 1. There are thousands of chemical reactions taking place in our cells every second, and that means that things need to get into the cell and waste products need to get out of the cell. How it does that is through this process of cell transportation. And that's what we're going to look at today. How do these molecules get in and out of our cells? Here is a typical cell membrane. The cell membrane surrounds all of our cells. Now let's take a look at some of the structures here. We see that it forms two layers of phospholipids. And so this is known as the phospholipid bilayer. There's two layers, bilayer. There's two layers of lipids. And so these little round things that you see, that's the glycerol, and there's a phosphate group attached to that. And we have two fatty acid chains that are sticking down from that glycerol. The round part is said to be hydrophilic. In other words, that it wants to be in contact with water, whereas the two tail regions are hydrophobic. They don't want to be in contact with water. And so the best way to arrange themselves is in a structure like this, where the tails are facing one another, where they're not in contact with water, and then the head regions are where the water is, inside and outside of the cell. We also have various, almost like tunnels, we're going to call transport proteins. And these are embedded in the cell membrane. Here we can see one, there's another one over here, we got another one over here, and so on, in which molecules can go in and out of. We also have, attached to some of these transport proteins, are other molecules. Now, if you take a close look, we can see a nice hexagon shape. That usually indicates some type of a monosaccharide. So these are what are known as glycoproteins. All right, so a glycoprotein is part sugar, glucose, part protein. These are going to be acting as receptors for the cells, which we're not going to really be getting into a whole lot of discussion on that. So let's take a look at how things can get in and out of the cell. Here's a wonderful drawing that I made showing the phospholipid bilayer. So here we have our two layers of lipids. There's one, there's two layers. Here's one of our proteins, our transport proteins. There's another protein as well. And let's just say that this is the outside of the cell, and this is the inside of the cell. And let's start off with diffusion. So this is the simplest type of cell transport. Diffusion is the movement of solids and gases from areas of high concentration to areas of low concentration. And you may need to pause the video here to write that down. Let's look at an example of this. Oxygen is something that we need. We have a lot more oxygen outside of our cells than inside of our cells because as the oxygen gets in the cell, it's being used up. All molecules have a certain kinetic energy. They're all bouncing into one another and they're colliding off of one another. Because these are such small molecules, these oxygen molecules have the ability then to go from high concentration areas to low concentration areas. So in this case, it's going from outside the cell to inside the cell. But it's not just a one-way thing. Here we have carbon dioxide. This is something that we make as a byproduct. We have a lot more carbon dioxide inside our cells than outside because our blood takes it away. So we set up this difference in concentrations. More on the inside of the cell, fewer on the outside of the cell, and so by diffusion, these molecules go from an area of high concentration to an area of low concentration. So it's not a matter of going in or out of a cell, it's looking at where are there more, where are there fewer. Now, very similar to diffusion, in fact, it's actually a type of diffusion, it's called osmosis. This is the movement of water from areas of high concentration to areas of low concentration. Notice the only difference in our definition here is that we're looking at water instead of solids or gases. And so let's draw some water molecules here. Oftentimes we may see that there's more water outside of a cell than inside of a cell. That sets up a concentration difference. And you guessed it, the water is going to move into the cell. It's going to go from an area of high concentration to an area of low concentration. But remember, we said that the tails of the phospholipids are hydrophobic. They don't want to be in contact with water. So how is it that water can just pass through the cell membrane? Well, it wasn't until a few years ago where scientists discovered something called an aquaporin. This is a very tiny little transport protein, and there's hundreds of thousands of these on every cell membrane that allows for water to go either in or out. It just depending on what the concentration is. Because we could have concentrations higher on the outside of the cell or higher on the inside of a cell. And when we have something like that, we can set up different types of solutions. We're going to look at three different types of solutions. You're looking at is a beaker full of water. Let's just say that this is 100% water. And then we add a drop of blood. Now blood is 99% water, about 1% salt, actually a little bit less than that, but keep our numbers simple here. Water is always going to go from an area of high concentration to low concentration. So guess which direction the water is going to go in a hypotonic solution? It's going to go into the cell. It's going to go from a high percentage, 100%, into a lower 99%. Now what happens in a situation like this is that water will enter the cell, but these cells are so fragile that they just burst right open. Here's an example of what a normal red blood cell looks like. It's a biconcave disc, kind of looks like a Smarties candy. This is what it looks like when it's in a hypotonic solution, and this is right before it bursts open. Now in a hypertonic solution, we might have a beaker of water, but this time, let's say it's a 5% salt solution, which means it's 95% water. And then we add a drop of blood, which is still 99% water, 1% salt. Guess what direction the water is going to go now? You guessed it, this time it's going to leave the cell. It's going from an area of high concentration, 99%, to an area of low concentration, 95%. And so water leaves the cell, and ultimately the cell is going to shrink. So again, here's our normal red blood cell. This is what it looks like in a hypertonic solution. So in an isotonic solution, we have a beaker that has 99% water, 1% salt. We add a drop of blood, which is exactly the same. Now which direction do you think the water is going to travel? You probably weren't thinking about it like this. You see, water is constantly moving. So that means that it's going to be going in and out of the cell at exactly the same rate, which essentially means, though, that nothing happens. The cell stays exactly the same in an isotonic solution. If you wear contact lenses, look on your bottle of saline solution. It'll say that it is a sterile, buffered, isotonic solution. It has exactly the same salt concentration as your tears do. Otherwise, the cells in your eyes would either swell up if you didn't have enough saline, or it would shrink up if you had too much. Well, that's only part one. That's the basics of cell transport. We're going to look at other types of transport in the next video. So I'll see you then.